So I was asked to talk about the mechanisms of antidepressant action and to approach it from both psychological and pharmacological, so biological angle. Next slide. And the question is really why we should explore the mechanism. And one thing is about, obviously, scientific curiosity. We really want to know how they work. We know clinically, as scientists are medics, we know they work. But it's really interesting to know how. But the second thing is also that we want to develop new medications. So the trouble clinically is that we've got very good medications and many people respond to them, but we still have about 1/3 of patients who don't respond to anything that we've got at hand at the moment. So through understanding how current medications work, we can get information about what is important in the biology of depression, what can be addressed, and how we can do it better. And I will try to see this train of thought in the development of medications over time. We also, this way we can get some more information about depression itself. So through understanding depression, we can plan how or design new medications, but also it can feed back. So through understanding how medications work, we can actually try to understand the condition a bit better. So it's also quite relevant. And how we can approach it, there's so many ways. We can go from this micro level of DNA, we can examine what happens at the level of the cells, we can look at the brain and the networks in the brain. We can use psychological lab and see how people think and what changes in their thinking and their perception of the world when medications are applied. And we've got a lot of tools. I wouldn't have time to talk about all the tools, but starting from the laboratory, through scanners, there's a lot of fantastic ways of getting information these days. Okay. So just to mention that we haven't had antidepressants for a super long time. It's been only really 70 years when we can help people who suffer from it. And the first antidepressant was imipramine, which was actually discovered by accident. So it was a medication that was developed as an antihistaminergic drug. There was attempt to use it in schizophrenia. It failed. It was almost abandoned, but then it was noticed that one person, one woman, really improved massively in terms of her depressive symptoms, and it was a big breakthrough. So then this medication was trialed and eventually marketed in, I think it was 56. And this is how medication history started. And then we started thinking, smart people started thinking about new medications. So I know that I was told I should address quite a wide range of people, so from undergrads to PIs. So forgive me if some things may seem quite basic for some, but actually I hope everybody will find something that will be intriguing and interesting here. So I couldn't not mention the basic mechanism of antidepressants as we think of them. So the one that relates to monoamine depletion hypothesis of depression. So there is this hypothesis that depression is caused by the low level of serotonin, norepinephrine, dopamine. And logically what we should do, what imipramine does as well, is to increase the levels of those. So this is how some medications were designed and how they seem to work at the biological level. So some of them, they inhibit reuptake of those neurotransmitters. So there's more of it available at the synaptic cleft. And some of them, monoamine oxidase inhibitors, they prevent the breakdown of those. So again, we've got more available to act. If I had a pointer, I would show you this TCA, so this tricyclic antidepressants. That's again is the evolution, how we thought about making the drugs better. So imipramine is quite non-selective, so it will be acting on what we want. It will increase serotonin, it will increase norepinephrine, but it also acts on a lot of other receptors, causing a lot of side effects. And actually the problem of those drugs that are very effective drugs clinically, but the problem is also quite a narrow therapeutic index, so the dose that is therapeutic and lethal is quite close, so there's quite a lot of problems with safety, giving a lot of medications to people. That sort of triggered people to think about how can we make it better, and that led to the development of drugs that will be selective. So we've got SSRIs, we've got NRIs, SNRIs, so drugs, you've heard the name, I'm sure, escitalopram, sertraline, that are selective reuptake of serotonin inhibitors. NRIs like reboxetine that act on norepinephrine transporter. SNRIs, so serotonin and norepinephrine transporters, venlafaxine, duloxetine. They also come with their own problems, so they are not free from side effects, but they are definitely safer to use. So the dose, suicidal dose is much different to what is therapeutic. And we've got bupropion that acts through a different mechanism. It actually targets dopamine transport. Then smart people started thinking how to make it even better, because we still had people who don't respond, and that's, again, a big problem. So people started thinking how we can tackle different systems, monoamine systems in different ways. And that led to the development of drugs such as mirtazapine, that's quite commonly used, that actually goes for alpha-2 noradrenergic receptors, and this way it causes increase in serotonin. So it's quite a lot of knowledge about biology going into this. Drugs like trazodone, so we've got inhibition of the transporter of serotonin, but also inhibition of serotonin 2A receptors that was thought to prevent certain side effects. And side effects like sexual side effects, they're a big problem. People stop taking medication because of them. So it's a big thing to actually to be able to get rid of those. And then, probably like 20 years ago, people thought about even more complex actions like vortioxetine here at the bottom. So you can see there's a lot of agonistic, antagonistic action. So it's really a lot of thinking going about how to put it all together to make things work. This is a nice slide. I'm not going to take you through all of this, but if you wanted to refer to it, it really nicely summarizes the action of different medications on transporters or receptors, and also talks about something that I will mention a bit later, neurogenesis. That is a very important aspect of antidepressant work. Now, you probably heard there was this article, umbrella review, that actually questioned the monoamine or serotonin hypothesis of depression. And I would like to paraphrase Mark Twain's that reports of my death are greatly exaggerated, and it speaks serotonin. And I thought, well, I could talk a lot about it, but it's not really the place. But I just wanted to mention our recent study that actually was really timely published right after this meta-analysis. And we're able to show for the first time that it was the first direct assessment of serotonin release capacity in people with depression. So we gave people amphetamine, it releases serotonin, and we were seeing whether the difference between and after amphetamine challenge in people who are healthy and who are depressed is different. And we are able, using PET, to show that actually there is a big difference. So people with depression have much lower capacity to release serotonin. So there's definitely a lot of space for a serotonin hypothesis, and we know that medications work. I was quite surprised to see this like shocking, like big title, quite dramatic. The chemical imbalance theory of depression is dead. I think it was Guardian. But luckily they say that antidepressants still work. Sadly, I have to say, I had a lot of patients coming to say, oh, I heard medications don't work. And it's like, but you're better. So it caused a bit of a stir. We have to fight. I thought I would talk about some other mechanism of antidepressant action in the context of a known effect, a known phenomenon of typical antidepressants. So typical, different to ketamine that was introduced quite recently. So this phenomenon is delayed clinical onset. We sort of know that the medications, they start working very quickly. So there's this burst of serotonin definitely within hours. We really see biological effects, but somehow people don't respond very quickly. There was a study, like an old study now, by Matthew Taylor, who looked into this phenomenon. And actually there is some response very early on, after a week or two, but it's not clinically relevant. People really feel better after a longer time. So through trying to understand what happens on the way between application of medication and then the clinical effect, we can understand a bit more about the mechanism of action and maybe then target actually things that are on the way to this clinical effect. So maybe we can think about other antidepressants that will target what is relevant there. And that actually takes us to another aspect of antidepressant action, so 5-HT serotonin 1A autoreceptors. When we've got a lot of something, in this case serotonin, quite often we've got this feedback mechanism telling the neuron, stop producing it because we've got enough. And in a healthy brain, that's how it should work. In the depressed brain, we actually want this increase, so this is not a great action. This led again to the development of medication that would be a partial agonist at this receptor, so basically stimulating it but less. And at the same time, it inhibits transporter of serotonin. So we've got more serotonin, and was thought that this way, desensitization of the receptor that takes time and it explains the delay in action, it would happen much faster. Unfortunately, this doesn't seem to be a super effective medication, so it's not much on the market, and also this quicker burst of increase in serotonin, people just couldn't tolerate it, so there's more to it than just biology, but it's a very important mechanism of how drugs work. There are other medications like buspirone that share this action, some antipsychotics as well. Okay, and moving on, I thought I would go back to neurogenesis. So there's a very important mechanism, again, potentially explaining why we've got this delay in typical antidepressant action. So I won't have time to go very much into detail, but basically, depression, chronic stress, they lead to atrophy, death of neurons, fewer synapses, lower levels of BDNF, and this is reverted by antidepressants. I will mention, when I will talk a little bit about ketamines, I will mention the difference between the two. But it is thought very much that typical antidepressants, they work through increased expression of BDNF gene, so it takes time, and then we've got increase in BDNF and the processes that follow. While ketamine is supposed to actually to just increase release, and some people attribute its quick action to this. Okay, so. And now I would like to move to something that maybe some of you didn't hear. And I think that's a fascinating area, which is combining psychology and biology. So like 30 years ago, basically psychologists were here, psychiatrists were here, and nobody wanted to talk to each other almost, but we are serving the same patients. This gap has been closing, which I'm very pleased to say, because it also fed into neuroscience and our understanding of the processes that happen in depression and when we give people medications. So I think we're at the level of shaking hands now, rather than a box game. Before I can talk about medications, I will just mention this very important phenomenon in depression, which we probably all know. So everybody had a bad day, and when we have a bad day, everything feels a bit like we are not happy, we are not enjoying things. This is a permanent thing in depression. It's called negative bias. So people will be picking up information that is negative, even if they get a lot of positive information, as in this picture, like a lot of positive stuff, but actually, well, you're not a nice person, it's like, oh, I'm a failure. So there's this thing, picking up and catastrophizing, exaggerating. The brain is very busy with this stuff. It's very much about glass being half empty in depression, but technically, it's a secret, but you know that technically the glass is always full. It just depends how we name the substance in it. But depression sees it as always half empty. And what I find fascinating, so we've known this phenomenon for a long time, like Beck based his CBT therapy approach on this, changing negative bias. And through development of scanning and neuroscience, we know that actually this is rooted in the brain. So I will keep it very sort of simply. It's a very complex system, but I think we can simplify it to like two elements. So we've got elements including the amygdala, very sort of, I don't want to say primitive, but very early elements of the brain structures that are very important. We share them with animals. They have to respond to threat, to fear. If there is a wolf chasing us in the forest, we have to run. We can think about it. So that's important. But the problem is when these structures start talking a little too much and responding to everything around. As humans, we've got quite a nicely developed prefrontal cortex that should correct it and put it in reality. In depression, we've got either overactive amygdala and related structures, or hypoactive prefrontal cortex, or both. Anyway, that leads to the state in balance. The brain is prone to process emotional information this negative way. So we've got the bias. And that's really interesting. So our team has been working quite a lot of trying to understand what happens. There's a lot of tests that can be done. We can put people in the scanner and look at their brains. We can expose them to social cues. Faces are very good at it because we react to faces. people will pick up emotion, people with depression, negative emotion faster when we've got those more faces as at the bottom. They will remember more negative self-descriptors than people who are healthy. And we can use all of this to actually to test the effect of medications. So what is a very interesting thing, medications act very quickly on those bias. So before we can see any change in the clinical symptoms. So if you look at those few pictures, medications through different classes, citalopram, SSRI, SNRIs, the dopamine reuptake inhibitors, norepinephrine reuptake inhibitor, they all seem to have the same effect after just a single dose to change how we perceive the world. This was done single dose in healthy volunteers, all those studies. But we can see that people actually see happiness a bit earlier in those more phases. So when we've got ambiguous phases, they actually start seeing positive emotion faster than before they were given medication. That's really interesting. That happens also in short, 7-day, 2 weeks, and longer treatment, so 6 weeks, we can see exactly the same effect. So I'm sorry, like without the pointer, but if you can see that misclassification of phases as set gets much better after already 2 weeks of treatment, and definitely then after 6 weeks. And people are better at accuracy of assessing the balance of emotion, and that applies in this study to surprise, interestingly, and happy, but not necessarily to set emotion. It may be all about the balance between how we perceive negative and positive information rather than about processing just one emotion, so sad or happy. And this effect is also seen in the brain. So again, a few studies just to show you that acute administration of medications, 7 days, a few weeks, it basically has a very similar effect. So we can see that structures are responsible for emotional processing. This bias changes, so they stop being so active in response to negative information, so to keep it very simple. This actually fits into the delay of antidepressant action, and Kat Harmer has been okay, all right. Kat Harmer has been working on this and trying to tell us that it may be about very early effects, and then we've got a learning process that needs to happen for this change in bias to actually to be enacted. That was in one study that showed that actually social interactions, they are very important. So people who improved in bias processing in terms of recognition of happiness, they did better in terms of depression if their perceived level of support was better. And I can just tell you we've done a few studies. I will go very quickly through this because we've got a minute. So this early change actually allows us to predict the response in 6 weeks. That seems to be very relevant to the mechanism of antidepressant action. And it's different to placebo. Placebo doesn't induce this change, so only people on the citalopram, their level of brain response, it goes back to healthy people. And I just wanted to show that it's also about networks, not just about one structure of the brain, it's about networks of structures that can predict response, so probably this communication. So in this case, it was like responders had a greater connectivity between prefrontal structures and DMN, that is the network that holds people in their brain, overactive in depression. So this better control may be relevant to future response and to how medications work. And just to mention ketamine, it made the cover of Time magazine. It is supposed to act in a very different way. The most prominent hypothesis is of disinhibition of glutamatergic system. So basically it works on GABA interneurons, so we stop inhibition. Pyramidal neurons, they have this burst of glutamate, then actually stimulates very quick release of BDNF. And as I mentioned before, neurogenesis is very important. Here it happens very quickly and acts. There's questions about whether it's about ketamine, which is a racemic mixture of 2 forms, or it's about one form being better than another, so-called R-ketamine and S-ketamine, whether it's really about NDMA receptor, AMPA receptor, because the metabolites were shown to act on their own as well. And we can see a very nice spine, much better with ketamine than under stress. And that I mentioned. There's some controversies, again, because it seems that naltrexone can abolish the effect of ketamine, so maybe it's about opioid receptors. And this is just one slide, my final slide, and I mentioned there is new drugs. I'm not going to talk a lot about it because we've got Gary Gilmore from Compass talking about it for the whole session, so you'll hear a lot about the mechanism of action of psilocybin, but mushrooms are back. Yeah, it's a summary of her. Like, we talked about different levels of action, biological, psychological approaches, and a bit of typical and rapid-acting antidepressants. Thank you so much for listening, and thank you, BNA, for having me here.